Today, we are going to be studying the process of meiosis using the compound microscope. We have three objectives. The first objective is to see all the different stages of meiosis. The second objective is to compare the different stages of meiosis to each other to see the differences. And the third objective is to understand why the process of meiosis is necessary. Let's summarize the theory we learned in class. Remember that meiosis reduces the chromosome number by half. To do this, we have two divisions. In the first division, your homologous chromosomes separate, which already halves the chromosome number. And in the second division, each of one of those chromosomes has chromatids which separate to halve the chromosome number in the final division. In this PowerPoint slide, you will see microsporogenesis. This is how microspores are produced through the process of meiosis. So, within the anthers of a developing flower bud, we have microspore mother cells. Sometimes we call them microsporocytes. These undergo meiosis to produce 4 microspores. Each microspore will further divide by mitosis to produce a 2-celled pollen grain. On the other side of this slide, we see megaspore production, which is termed megasporogenesis. Within the ovary and ovule, the megasporocyte divides by meiosis to produce 4 megaspores. Only one of these survives and gives rise to an embryo sac. Inside that embryo sac, the female gametophyte will divide by mitosis to make 8 cells, 3 antipodal cells, 2 polar nuclei, 2 synergids, and an egg. In an angiosperm, which is a flowering plant, a double fertilization will occur, where one sperm from the pollen grain will fertilize the egg, forming a zygote. The other sperm fertilizes the fused polar nuclei, forming an endosperm. The zygote will then develop into an embryo and then into a mature plant. We will now view the different stages of meiosis in the male anther and the female ovary of the lily. Here we are looking at meiosis in the male anther. We see the first early stage of prophase 1, which is known as leptotene. The chromosomes are thread-like and all over the nucleus, and they have already replicated. So now we're on our second stage of early prophase 1, called zygotene, where your chromosomes have replicated and now they started to pair, where they zip together so that they can go on to the next stage for crossing over. And here we go on to our third stage of prophase 1, which is called pachytene, where crossing over is taking place. And you can see in some cases the chromosomes are really close together for the crossover to occur. Here we see the fourth stage of prophase 1, which is diplotene stage. Crossing over occurred during pachytene. Now we see visible evidence that crossing over has occurred, and it is a chiasma or chiasmata in plural. The chromosomes are also starting to pull apart. And this is the fifth and final stage of prophase 1. It's called diakinesis. At this stage, the chromosomes are at their absolute smallest and getting ready to move on to the next stage. We are now on to metaphase 1, where your homologous chromosomes, called tetrads or bivalents, are getting ready. You can see the spindle fibers are arriving to align themselves on the metaphase plate. You can see it clearly in these 2 cells. This slide shows us late anaphase 1. You can see the chromosomes have separated. You can see them separating here. And here you can see going into early telophase 1, which we see there. And by here, you actually see telophase 1, end of telophase 1. Here we see a slide that shows late anaphase 1. And you can see in this cell here, it's shown very nicely. The chromosomes have separated. And we can also see moving on to telophase 1 and actually moving a little further onto telophase 1. In this slide, you can see that we've come to the end of prophase 1 in that we can see an example of telophase 1. We see telophase 1 there. And then the cell next door, these cells have moved on to prophase 2. In this slide, we see metaphase 2. So you can see 2 cells lying next to each other and the chromosomes beginning to arrange themselves on the metaphase plate. We have finally reached the end of meiosis 2. This is called telophase 2, and we see we have 4 cells that have been formed. These cells, in the case of the anther, are called tetrads. Each tetrad is actually a gamete, and we actually call them microspores, and these microspores will eventually develop into pollen grains. We are now looking at meiosis in the female. We're looking at a through section through a very young developing ovary. Ovules are beginning to develop, and an enlarged cell called a megasporocyte can be distinguished. You can see them there. In this slide, we see we have come to the end of meiosis. So at the end of meiosis, we would have had 4 cells. 3 of them degenerate, 1 of them divides by mitosis to form 2 nuclei. These then go through a second mitotic division to form 4 nuclei. In this slide, we see the 8 nucleate stage. So those 4 cells we just saw have divided by mitosis again. So now you will have 3 antipodals, 2 integuments, 2 polar nuclei, and 1 egg. We have finally come to the end of female meiosis, where you saw the egg in the last slide. That egg was fertilized or will be fertilized by a sperm cell, which will form a zygote. The zygote is not visible here. However, the other sperm, because this is a double fertilization, will fertilize the 2 polar nuclei, which you do see here. So this is a triple fusion nucleus. And this concludes the video on the process of meiosis.